BTEC Applied Science for AAQ, and this is a biology video. It's the last one for section B. I'll be going on to section C next, and this is about gene expression. We've been talking about DNA and the structure of DNA. Now, gene expression, what's that then? Well, now we know that almost every cell in our body contains exactly the same genetic information, yes, which is about 20,000 genes. Now, if you were a cell in the heart, how does that DNA know that it wants to make proteins needed for a heart? If it's in the stomach, how does it know that it's supposed to make proteins needed to do like digestion stuff in the stomach? How does the DNA in a heart muscle cell know that it should only end up making proteins needed for that particular job? Because basically every cell, in every single cell in your body, there's all of the genes needed to make you. Yes, for every single part of you. But in any part of your body, you don't need all of that. So for any particular application, genes need to be switched on and switched off. On if they are needed, and obviously off if they are not needed, which will be most of them, most of the time. And that's called gene regulation. And gene regulation controls gene expression. In other words, if the gene is actually switched on and does something and expresses itself. So gene regulation controls gene expression. Now, cells control which genes are expressed, when they are expressed, and how much of different proteins are actually made. At some time you may need lots of a particular protein, at some time you may need very little or none of it. And the cells control that. There's lots of different ways that it is controlled. It's mostly controlled a little bit like peer pressure. What happens in a cell is based on the signals that it gets, chemical signals from its surroundings, and that usually means the surrounding cells. So, for instance, in the middle of this little gang of lads here, he's a stomach cell, and his neighbours are telling him, make some protein that will make stomach acid, or help digest this Mars bar that I've just eaten. Okay, so it's mostly from chemical signals that it gets from its surroundings, from its environment. Now, gene regulation can actually take place at lots of different stages. Do you remember transcription? Yes, and that's where the bits of the DNA are copied. And then do you remember translation, which is when the ribosomes actually make the protein? So you start with your gene, which is a section of DNA, and then it may or may not be transcribed. And there's clever ways for turning on transcription or not. After it's been transcribed and you've got your messenger RNA, then there's processes there which can determine whether it actually carries on or whether it's just broken up because it's not needed. In translation, there are processes there to determine whether it is translated or not. And then even when it is translated and you've got your protein, there are lots of regulation going on there to see if that protein actually goes on and does stuff. So at lots of different stages, then gene regulation takes place. Okay, transcriptional regulation, regulatory proteins, or transcription factors they are called, control transcription. This is one of the ways that it's done. And what they do is they bind to a particular part of the DNA, and they might promote transcription or they might stop it. Yes, so basically the message would be, come and copy this bit of DNA, or don't copy this bit of DNA. And a similar process can occur after transcription and before translation. And proteins can bind to the mRNA with instructions to either promote or stop translation. So binding proteins which carry messages, copy me, don't copy me, translate me, don't translate me. This is one of the mechanisms. Now, the mRNA can be altered after transcription, and bits and pieces of it can be removed depending on what the cell needs, or it can be completely broken down. Okay, so clever proteins can attach to different sections of it, and they can remove sections, or they can just break it down completely. This is interesting. We used to think that until relatively recently, that most of our DNA was useless because we only figured out what 0.2% of our DNA actually did, yeah, which was carry the genetic information. However, and the rest was called junk DNA. One idea was because we evolved from species which are now completely extinct, that it just happens to be there because our ancestors, the species before us, had this DNA and we inherited it from them, but it doesn't actually do anything. It's now understood that a lot of this so-called junk DNA isn't useless, this non-coding DNA. It actually contains all of the information that I've just been talking about, yeah, to control gene regulation and to control gene expression.